Joana, Prata, talking now, giving a keynote lecture about the impact of airborne and foodborne microplastics on human health. And then we will break for half an hour. So, Joana, the floor is yours.
Thank you so much. Thank you for the invitation to be here today. And I hope you can handle this time as well a little bit. Let's try to
We can
keep
handle
it.
everything. We can handle everything.
Yeah, of course. And so my presentation is about human health and mostly what we don't know. That's why we're here today, right? And this is something that you already know, of course. I just wanted to point out that the plastics we use them because there's no other material that has the same qualities. And when they showed up, it was presented as a novel material that would save the world and do all these good things. And now we see that it's not the case. Of course, there's always benefits and costs in doing something like this. But there is this problem of material development because we cannot substitute it. It's unique, it's lightweight, it's flexible, transparent. All these qualities are very good in plastics and have been very helpful in many applications. So it's very difficult to not use them. And as you know, most of them are used in packaging, which is kind of a short-term application. Of course, for food safety, it's very important, the packaging. It's saved us a lot of food waste and also a lot of foodborne illnesses that we could have had without the plastics. But now we have this problem to deal all the waste. Then we have other applications, and of course some of them are more long-term, for instance in buildings and cars. These plastics will stay a long time in those applications. They are also a problem, but maybe the ones that are generating a lot of trash, we should deal with it first. And in the world, it's still growing, the use of plastics. So as you know, most of them are not recycled. Only a little bit has been incinerated. So most of them, since we started production, are still here in the world, and we are keeping adding more and more plastics. Some of them are in landfills, but still landfills can release microplastics, and some of them are in the environment. So this is a problem that is still growing. And this was a bit of an introduction. Of course, you're familiar with it. I just pointed out that it's very easy to see in any beach, and Portugal has beautiful beaches, but you can still see the plastics, the fragments, and the pellets easily in the beach. Of course, as you've known, we talked about a lot about this. Microplastics are a class of contaminants. We cannot talk about one microplastic because they are all different. And of course, in the lab, we can have different polymer types, but when we go to environmental plastics, they are very different from each other. I would even say they are unique. They are like us. Each is their own thing. So it's very difficult to assess their properties and their toxicity because really they are very unique in the weather patterns, the size, the shape, the color, all that. So maybe to do the modeling, as we talked yesterday, we should focus on how these factors affect the behavior of plastics, either in the environment or in toxicity. So we need to narrow this down because otherwise it's so much to test, it will be impossible to reach a conclusion. And also, as we've spoken so much about this, it's the size, the problems with different size ranges. And here you can see that if we are using a Mantatrol net, that goes for about 333 micrometers until the 5,000 micrometers. And if you compare what you get with going from 1 micrometer to the 5,000 micrometer range, in this smaller range, if you go smaller, you get 40 times more microplastics. So it is a big difference. Of course, we have all the other variability as well with the methods. But just the size, just looking at this is a big variability between the results. That is why it's so difficult to compare between studies. And if you look, this is some sampling I've done with fish and cats and dogs, and you can see here that the smaller sizes obviously are the most abundant in tissues. Also in the environment, we expect them to be more abundant. Of course, it's very difficult for us to go here because the methods available do not work well in these very small sizes. But you can see that for biological matrices, over 80% are under 20 microns. And this is expected because they have to cross the tissues, and the bigger they are, the less likely they are to get into the tissues. I don't need to remind you of this, just pointing out some things in this part, which are the different microplastics, the different sources. Of course, the fragments here today, we are having microplastics being fragmented from the chairs, from our clothes, anything that is polymer. And that is hard to address. Of course, maybe in production we could do something with plastic design. But then we also have applications like microbeads in cosmetics, or the way we manage pellets. So these are different approaches. We have to do different things, because with these industrial applications we can go and ban them or regulate the way they are handled. But the ones that are being released from our clothes, from the objects in our homes, it's more difficult to address. So we'll need to have different strategies. And of course, all this gets into the environment and eventually gets in our food, because we are top consumer, right? We are at the top of the food chain, so we are exposed to all of this. And we have found them everywhere: salt, mussels, fish, sediment, water, anywhere you can find them. They are vectors for microorganisms, which is a bit worrying for human health, because they can transport pathogens, they can cause small lesions, open a door for the pathogens to get into our bodies, and also the way they act with other chemical contaminants. It can be good, it can be like a sponge and do not release it to the human body, be like a sink for chemicals, but it can also work otherwise, and they can get into the body and release all those chemicals inside us. So I think the question we are all asking is, are we exposed to microplastics? And we already know from all these talks we are definitely exposed, although we don't know how much we are exposed so far, because there has not been enough research on this part. And I think this is where we have to start, because if we don't know exposure, we cannot conduct a good toxicity testing based on this exposure. We need to know how much, what type of plastics, the sizes, the shapes, and all that, so that then we can go to the lab and do the tests. And here I try to focus, because I'm a veterinarian by training, so I try to focus on animal products, which also translates well what is going on in the terrestrial environment. There has been a lot of research for the aquatic environment, so most estimations are based usually on fish, mussels, and all that. But for the animals, we most eat meat, right, in the world. So for these animals, there's not a lot of content yet. And for fruits and vegetables, even less. I know we talked about it yesterday, but it's still very little research on this. And to estimate all the different things we eat, we have to do a big list of different foods, and ideally using the same methods. Because as you can see here, the ones that we have data for have very wide ranges, because I took this data from different studies and they use different methods, different cutoff sizes, so we get a large variation. And as you can see, these are two models. It's not really models, it's the way you calculate daily exposure for the World Health Organization and for the European Union. And we have a lot of missing data here. Pig meat, we don't know. Bovine meat, we don't know. Liver, kidney, other things that are not only meat. And even meat, if you go to bovine, to small ruminants like goats and sheep, sometimes you have a little bit of information, but it's very scarce. So we really don't know what we're eating in terms of microplastics. And of course, it's not easy to conduct an estimation. Here we can go for the available data, which is very limited. We think that we can go up to 15,000 just in meat. But then we also have air exposure, which is estimated around 3,000 particles, and then the water, the fish, and all that. If we add more, 30,000. But this is of very big sizes usually, because the methods are limited to go to smaller sizes. And the water, as we've seen, is also contaminated. Even the lab water, we think, oh, we're using distilled water or ultra pure water. It also has microplastics, so we really have to filter everything. And so when we're at home and drink a little bit of water, we'll get some microplastics in our drinks. But if you think you're in this beautiful country, Italy, and you want to drink a little bit of wine, I'll just pass it to the wine. It also has microplastics, so you're not, you cannot win with this. But going back to the water, we found it in natural waters. Aveiro Lagoon, which is already a mix of with salt water, but also Voga River. This was a very in the countryside, so it was less contaminated. And then when you look at the Douro River, this is near Porto, so it's almost at the sea, at the estuary. You can see this side is the uphill, and this is the downhill. You can see if you go to the countryside, you find very few microplastics, few fibers. And then if you go here, there is a big wastewater treatment plant. You find a lot of microplastics and fibers that are coming, we think they are coming from the wastewater treatment plant, especially because in the small river, they release the wastewater to, is higher than the surrounding areas. And then the boat repair, because as we talked about yesterday, the cores of the boats, the paints, all that can also release microplastics. And also there might be an influence from getting downstream and closer to the high density. And the wine, as we said, microplastics as well, so you cannot drink anything. And fish as well. This is try to gather information about fish across the world. And as you can see, the orange and red are where the fish are most contaminated, and almost all of them are orange or red. So this means that 50 to 70% of fish presented microplastics, mostly in the gut, because studies usually focus on the gut. But as you can see, most of them already have microplastics anywhere in the world. And we looked at fish in Portugal because we eat a lot of fish. We are near the sea. And so we eat these fish, tracurus, and we tried to find them in internal tissues to see if they were moving around. Of course, in the gut, we could find the biggest ones because we also included the content of the gut. But then we looked at the heart. We could find very small plastics. We tried to use the heart to be an example of the circulatory system because this fish is very small. To collect a blood sample is very difficult. But the heart as a proxy for the circulatory system, the kidney as well, and also the gills. The gills are exposed to the water, so it's very probable they get contaminated. And as we've seen before, most of them very small, 1 to 10 micrometers. Of course, this is with Nile red. These are suspected microplastics. We could not confirm them. And when we tried to go into microrama to get a reading on this, if you hit these very, very small particles, they will just fly away or get burned on site. So it's very difficult to get the spectrum of these particles. And what happens when we ingest this fish and wine and water? What happens in our body? Well, I tried to summarize this here. You have more details on the paper if you want. And basically, the microplastics reach our gut, and then they have this mucus layer they have to pass through. So some of them will pass through this layer, and not very many. It means that at every stage we'll lose microplastics. So some of them will be able to cross the mucous membrane and be near our cells. These are the intestinal cells. We have enterocytes and M cells, and also some macrophages that are around. And so we think that from all the research, I tried to gather all research done before in the 70s and 80s, because at the time we thought that it was a good idea to use microplastics, which at the time we called microspheres, to deliver pharmaceuticals to parts of the body. So there's actually some studies from rats and even from humans where we expose them to microplastics. So if we go back and gather all this evidence, I tried to bring it back, we have a little bit of data of what is happening in terms of pharmacokinetics. So I tried to summarize it here and all the other slides on this. So most of the data show that the enterocytes, transcytosis is the main route of transfer of microplastics in the intestine. Of course, the M cells also play a role, but it's not as important as we thought so far. And some can go in between the cells. Of course, there are openings if the adhesion connections are not very tight. Sometimes it opens and the plastic can pass through. This is the main mechanism for bigger particles. And of course, if there's an injury or the cell, when there's desquamation, the cell is changing, it can also have holes where the microplastics go through. You have the macrophages, they are the alert system, right? They are surveilling our body. and they also surveil our gut and can catch these microplastics and bring it inside to see what's going on. And these particles then can move into this interstitial space and eventually get into the pyre patches, which is a lymphatic tissue we have. And here in the lymphatic patches, they can also get caught up by macrophages, and eventually some of them can get into the bloodstream. Of course, this is a gradient, as I said. We think that less than 1% and maybe even less than 0.1% of the microplastics we ingested can get through, and even less will get through the blood. So there is a we lose plastics along the way, which is good. We don't want them in our bodies. But here, in terms of sizes, what we've seen from studies, we think there were some studies with inert particles made of starch, and these studies shown that particles up to 150 microns could get into the gut, be internalized in the human body. And then there are some tests. This study also used dogs, which they found the limit to be at 100 microns. And then there's another test with polyvinyl chloride in dogs, which confirmed the limit even for polyvinyl chloride of 100 microns. So it seems that even 100 micron particle could get in. Of course, this is in terms of probability. If you have a smaller particle, the higher probability that it will get through. If you have a bigger particle, it's very unlikely, but it can happen as well. The air is also contaminated. Everything, the dust you see at your home while you're cleaning, it also has microplastics and a lot of fibers. Of course, in the lab, we try to keep it clean, but even there we found a lot of particles. But then if you work in the laminar flow hood, of course, you can contain the contamination a little bit. It's never zero if you do enough time, but it's very low. And this is a preliminary result. We are trying to study in the university the deposition in different areas. And we tried to expose very low-tech approach. We tried to expose filters to the air and see the deposition on these filters, on these Petri dishes. And so we did only 4 hours because we tried to have it during the school time. But most of them were not different from the blanks, had a lot of blank contamination. Oh, the only one that was the library, probably because there's a lot of people coming in and out, and the books are really hard to clean, so it can accumulate fibers. And you can see for the library it would be 216 fibers per hour per square meter, and most of them we found them in larger sizes because we're just using a stereo microscope. But this means that our homes, it can also happen. And also when we're eating, there's also this deposition in our food, so it's another source of plastics in the food. This is an active sampling, so in this experiment we had a pump pulling in air, trying to simulate what happens when we're breathing, right? And it was in several houses in Aveiro, and we quantified the microplastics, the fibers, and what we thought there was synthetic fibers. So the sum of the microplastics and the synthetic fibers, we estimated 1.1 particles per square meter. And you can see here, it's a bit bright, but you could see here some particles, the fluorescence one that we found on the air. Of course, the air we're breathing, it's also contaminated, as we've seen in these previous results. So what happens is most of them are not going to reach our lungs. I think it's worse if they can reach the alveoli of the lungs, because that's where it's very thin, right? It's where we're exchanging the gases, so it has to be very thin and very connected with our circulatory system. So if it reaches that stage, it's worse because they can pass more easily. But most of them will get stuck just in our nose because they are very big particles and they just get stuck in our nose. Sometimes we feel them when we cough or sneeze and they go away. But if they are a little bit smaller and can get through, there are several mechanisms that we have and the way that we are built anatomically so that the particles get filtered out. It means that they will deposit in our trachea, the bronchi, and so we lose particles along the way. The biggest ones will be at our nose, and the smallest ones are the only ones that can reach the lungs, the deep lungs. And so for the lungs, you know this from particulate matter, right? The particulate matter below 10 micrometers in aerodynamic diameter are the ones more likely to reach the lung. And the smaller you go, the more likely. So for our bodies, we think that below 5 microns, 10 microns, it could reach the lung. Of course, if you have a fiber and it goes like this, and it goes perfectly all the way, it could reach the lung as well and be bigger, because it depends on the shape of the particle, the orientation, the way it behaves when it's suspended. So these particles, let's admit that only the below 5 microns, these are the ones that mostly reach the alveoli, they reach here. And just as in the intestine, they can get through the pneumocytes, the type 1 pneumocytes and type 2. Mostly these cells, they can get through and get into the interstitial spaces. Also, if there's a gap, they will also get through. And here, they can accumulate in the tissue or reach the circulatory system. Of course, here it will be very probable because there's a lot of blood and small capillaries, very thin, so there's the gas exchange. But I would also like to point out this. It's that even though we can only breathe in these very small particles to reach the lung, there are bigger particles in our lungs. Because when they get into circulation, in our intestines, for instance, if they get into circulation, they are big. If they reach the lung, the vessels are very thin and they will get stuck. Okay, there is entrapment of particles in the lung. So if we sample the lung, we'll probably find bigger particles there just because they were in circulation and got stuck in there, but they come from other places. And since now we are just doing digestion, we don't know if they are here, if they are in the capillary stuck, or where they come from. So we only know they are in the lungs. Another aspect very important in the lungs is one of the cleaning procedures of the lungs, which is the mucociliary escalator. Basically, inside us, in the trachea and the bronchi, we have mucus as well, and we have ciliary cells that are beating and pulling this mucus upwards until it gets to our throat, and we swallow it. This is happening to all of us right now. We don't feel it. And what happens is when we breathe in the plastics, as I said, they will get filtered out by our system and get in the trachea and the bronchi. They will deposit on the walls in this mucus. And then our body is beating the mucus upwards until our throat, and we'll swallow it. So some of the plastics that we find in the gut actually are in our breathing, right, in our respiratory system, and we swallow them and we expose our gut as well. So it's a bidirectional mechanism. But do we find microplastics in the human tissues? Well, you already know the answer to this question. Of course, we found them on all these tissues: lungs and airways, liver, colon, kidney, spleen, blood, placenta, feces. The big problem is that to deal with these samples, the human samples, you have a lot of constraints, have to have ethical approval, then to deal with these tissues as well, have to have a lot of biosecurity, so it's difficult. That's why usually you go for the easiest samples to deal with. For cats and dogs, because I'm a veterinarian, and you know, cats and dogs live with us in our homes where we spend most of our time, so they are very good sentinels for our home environment. So cats and dogs are very easy to get samples from. You just have people ask for necropsies for their cats and dogs, and you can just collect a little bit of sample and sample them very easily. And so we did this with cats and dogs, and you can see, for instance, here, polypropylene and polyethylene terephthalate that we found in the lungs of a dog. And we found that most animals were positive for the plastics, and 80 out of 240 samples of different tissues were positive. As I said before, most of them very small, under 10 microns. And we're talking about the sentinel role of the cats and dogs. Maybe a lot of you have pets at home and you have different behaviors with your pets, but are they really relevant and a good indicator for human exposure? Well, sometimes they are, sometimes they are not. It depends on how we do with our cats and dogs. For the respiratory system, we think that they may be a good indicator because they are smaller, they are closer to the ground where the concentration of particles are likely to be higher, and basically they are breathing the same air as us while we're at home. But for the food, maybe they are not such a great indicator. Most animals now eat pet food. It's also contaminated, but we don't eat the pet food, so It's maybe the gut exposure. It's not really good to tell of human exposure. Of course, sometimes we share something with them from our food, but since it's mixed with the other things that we don't eat, maybe not a good indicator for the food exposure. But for the air exposure, I think we should explore this. This is the a planaria that was exposed to Chironomos, mosquito larvae containing microplastics, because they prey on these larvae. And we could find some changes in the way, in some toxicity from this exposure, but also the most important thing that I would like you to see are these histopathology. We did slides to try to find the microplastics. And with polarizing light, you see that some particles are bifurcate, which is maybe helpful to find the microplastics in the samples. And we try to do staining as well, but it's not very easy when the particles are very small. But I think this is the way to go in the future. We really need a technique to be used with histopathology, because we need to know where they are. Are they inside the cells? Are they outside? Are they in the interstitial tissue? Are they causing an inflammatory reaction? We don't know, because we really need to see them like this in the tissue, interacting with the tissues to be really sure. But so far, it's very difficult to see them. And so what are the consequences of human exposure to microplastics? Well, we talked about what happens when they get in, but now let's talk about what happens after they're in our body. Well, as we've seen, that sometimes they can cross and get into the interstitial tissue, and sometimes even reach the circulatory system, and through this reach other parts of the body. As I told you before, many of them will get entrapped in the lungs, get stuck in the small capillaries. But this can also happen in other places in the body if the vessels are small enough. But the lungs, they really have small vessels and a lot of vessels, so that's why we find them more there. Also, there's some metabolism. I know this is a bit controversial, but if you look at these studies done with plastic materials and applications, for instance, for implants of plastic in the human body, they find degradation of plastics. And of course, we also, that one of the ways that our body deals with strange things, like foreign materials, is to release reactive species. So actually, we're creating oxidative damage on these microplastics from the reactions, the inflammatory reactions of our body. So if they stay long enough, they can get a little bit degraded. Also, if they are in our guts, there can be some interaction with the microbiota and get a little bit degraded. And the most important part that everybody asks about is the excretion. Are they going away? And I have good news. We think yes. They are probably going away in our feces. For the bladder, not so much, because they are too big to go through the renal system. If they degrade into additives and all the chemicals they release, this can be excreted through the urine. But the microplastics themselves are not very likely to cross because they have a very high molecular weight. So the way that the body deals with them, it's through the macrophages, basically the liver, cleans them, either the hepatocytes that can capture them and release them to the bile, or the macrophages pick them up, either in the liver or in the spleen. Of course, across the body this can happen, but mostly the liver and the spleen. They pick them up and they release them either on the biliary excretion, or directly the macrophages can go to the intestine and directly release them to the intestinal lumen. And this way they are found in the lumen again. Even if they did not come from the oral exposure, they will reach the intestine anyway. And they hopefully will get excreted in the feces. As you know, there is some enterohepatic circulation, which means that when we release something in the intestine, it can get absorbed again and go to the liver and get to the intestine again and be in this cycle. But hopefully they are released in the feces, even if they come from the air and into our lungs. So that's why we find so many in our feces. Of course, some were never absorbed. It's just we ate and they passed through and were released in the feces, and some got in our bodies and then were cleaned again to the feces. For the lungs themselves, well, we have found them in lung biopsies. As you know, they may cause the disease by this oxidative stress, if they move to the tissues, if they cause damage, but in the lung as well by dust overload. Basically, if you inhale so many particles, it can create a positive feedback loop. Your inflammation is so great because there's so many particles and the macrophages cannot clean them all. They start releasing cytokines and stimulating more inflammation, and it just keeps going, and your lungs are very inflamed. Of course, this is mostly seen in occupational diseases, not now, because thankfully now we have good ventilation systems, masks, we tell people to be very safe in their workplace, but if you look at the 70s and 80s, you actually had people exposed to these microplastics because they were doing paints with microplastics or working with microplastics, producing microplastics in their jobs, and they had a lot of dyspnea, airway and interstitial inflammatory responses. And if you send them home to get well, they would improve. If they come back to work again, they would get worse. So you know it's related to the exposure to microplastics. Of course, in our daily lives, we're not exposed to this much microplastics in the air, so it's unlikely that we get these very strong reactions, but there's this possibility. And of course, the toxicity pathways, we have already talked a little bit about this in the other presentations. Most of them seem to be related to oxidative stress. It could be from the surface of the plastics, also some of the things they release, and even the inflammation they cause, because they will stimulate the inflammatory reaction, which will also cause oxidative stress. Then there's effects on energy metabolism. It can increase energy consumption if you have enough inflammation. It can decrease feeding. Of course, for humans, we had to eat a lot of plastics, but if you're talking about smaller animals, we can have a decreased feeding if they fill the gut. And we can have gastrointestinal effects. For instance, they can interact with the enzymes in our intestines and even the nutrients we are trying to absorb, and they may not work as well. For instance, if they connect to the lipase, you cannot break down the fats to get absorbed. So there may be also some effects, nutritional effects of the plastics. Then on the immune function, we've seen that they sometimes stimulate immune reactions, sometimes they reduce, and animals are more likely to get diseases, but also autoimmune diseases. And this is very important because it's also connected how microplastics can cause inflammation, is that the plastics, they just go around, and sometimes they interact with some of our proteins, and they can damage these proteins. And if the protein is damaged, sometimes the proteins get stuck on the surface of the plastic and the inflammation is around it, but sometimes it just damages the protein, and then our body recognizes it and starts an inflammatory reaction. If it does so, sometimes we can start to have autoimmune diseases because we're reacting against our own components in our bodies, and sometimes you just get inflammation. It really depends. And then neurotoxicity as we've seen, the cytokines, the neurotransmitters, as we've seen in the previous presentation, and behavioral changes. We think most of this comes from the reactive oxygen species, and I think this is very important to identify the pathways which the toxicity is coming from. So even the neurotoxicity parts seem to come all from the oxidative stress. And these are some tests I've done with organisms. Here, I just like to say that because we use big microplastics, like 100 micron, that could not get into the animal, the zebrafish embryo. Probably the toxicity came from the leachates, but most importantly, were environmentally relevant concentrations. And we found that was not a linear curve of toxicity, because sometimes plastics interact with each other, sometimes they aggregate, sometimes they are so depending on the concentrations, they may behave differently and have different toxicity effects. So it's very important to go to the concentrations that actually are in the environment and do a range around that to see what is the effect. And also in Chironomos, the larvae, they can fill the gut and have all these different reactions, including different body lengths, emergence time. Sometimes it increases the time between the males and the female emergence, so it can have an effect. And finally, human health, One Health. As we've talked about here, there's a lot of impacts from indirectly, that indirectly will impact human health. For instance, the agricultural impacts in the habitat properties, reduced plant growth, all this is very important for our nutrition and our survival in the planet. So this indirectly can affect our health. And of course, we need everyone because it's so many problems to address. We really need to involve different people with different skills, because otherwise we'll not be able to deal with this. And now, just a thing that people always ask me, what can we do? And I just bring you an example. Of course, there are many things, from using less plastic in our clothes and all that, and cleaning. But I'll just give you an example of food. I just try to use less plastics. These plastic cutting boards that we use at our homes, you see them scraped, right? Each time we run a knife. We are producing microplastics every time we cut with this. And studies have shown that the chicken meat gets contaminated and goat meat from being cut in this board. So change your boards at home. And also the packaging. Takeaway containers, just opening a plastic bag, having chicken packaged in polystyrene, all this is releasing microplastics. So even the chicken can already come, the chicken meat can already have some microplastics inside it, but then you put it in a plastic packaging and it will be even more microplastics. So then this is already talked about, I will not get into details, but we need a better system even for waste management. And I invite you to submit. If you have any work on microplastics, you can submit. I have 2 special issues open. One more for animals, and especially for terrestrial system, if you have details on that, because there's almost nothing on that. And one broad one on monitoring microplastics in the environment. Thank you so much.
Thank you, Joana, for the very nice presentation. We have time for a couple of questions, and then we go for coffee.
Don't be shy.
Yes, go ahead.
The lack of caffeine.
Yes, please.
I am Salvatore Bello, University of Turin. When you were speaking about human effect, you spoke about feces and urine. We found in feces, but not in urine. This I well understand.
Yeah, we know that the urine production, the molecules that can go through the glomeruli in the kidney, it can only let molecules of very small molecular weight pass through. So you know that the microplastic has a very big molecular weight, and it's not likely that it will be filtered by the kidney. Of course, it can get stuck in the kidney, in the glomeruli, in the small capillaries, but it's not very likely that is a good excretion. Of course, sometimes it can happen if it just crosses over, but it's not an active excretion of microplastics. The active part is through the intestinal system. The macrophages and hepatocytes release all the litter to the intestine, and then it goes out in the feces.
Because we are speaking about microplastics, not nanoplastics.
Yeah, there are some studies, the studies I used from the 70s and 80s to study that, to do the revision and understand that, because I wanted to start this discussion of what is really happening in terms of pharmacokinetics, is that sometimes they used nano, sometimes they use microplastics. Usually they have a range of sizes, but all of them show that even humans, rats, dogs, they show different animals that usually they get excreted in the feces. So that is the pathway. That's why we find them in the feces. And maybe the feces can be a good indicator to the exposure in the future. So I don't know. I think we need to explore this a little bit more.
I can add some information about this because I am carrying out a plastic on epidemiology study in Italy, and I found microplastics and nanoplastics in urine with size ranging between 0.5 to 1 micron. So are very, very smallest particles. The biggest are captured by kidney because it's a filter. Okay?
Yes, exactly. The big ones can get stuck in the tissues, and if you have small enough, it will pass through. But generally, they are big.
And another problem is that compared to blood concentration of nanoplastics in urine, I found a minor concentration because during the transportation, nanoplastics are absorbed by other organs.
Yeah, exactly. They spread out. Even I know I had in the picture, but I did not talk about even to the brain. Sometimes it can get through the barrier, so they are just spread out through the organism. But generally, the clearance rate is quite fast. But I should add that we're talking about tests with pristine microplastics, right? Without anything on the surface. If we cannot extrapolate fully to what's happening with the microplastic covered in organic matter with all the other chemicals, all the functional groups, that it may be a little bit different from the pristine materials these studies used.
Maybe this means that they accumulate non-plastic in our body.
It means that we accumulate nanoplastic in our body. We don't eliminate that because if you don't find in urine, it's
nanoplastics have similar transportation as the pods. So minor is the size, major is the absorption in the organs. And the fat organs is better, the absorption. In fact, we are studying the correlation between nanoplastics in brain and minor depression.
Yeah, for the bigger microplastics, usually the bigger the particle, more easily the body will detect them and clear them out. And so for the bigger one, of course, you have half-lives, but for bigger ones, you can clear them out in a couple of hours. But the small ones can stay a little bit longer. And of course, we're talking about half-life curves. So you're tending to zero, but you may not get ever to zero. So there may be a little bit of accumulation. But so far, we've not seen the studies. We really need this type of studies, epidemiological studies, because in animals so far, there's not been a relationship with age with having more microplastics in the tissues. For instance, in the animals, we had 50 animals, cats and dogs, and the older ones didn't seem to have more microplastics. But they live also in very different environments, so it's not very easy to tell. So maybe a study also with rats would be good to do a big long-term exposure.
I will get two very quick questions with two very quick answers because we are running really late. So there's one there and there's another one at the back there.
Well, thank you very much for the very interesting talk. So you would say we are not only releasing the bigger particles, but also the very small ones? And what about if they're getting stuck or when they may reach the tissue or the bloodstream? Would you think it could cause cancer or strokes or anything?
Yeah, I think so. All these mechanisms, because of the damage, and if there is a prolonged damage in the tissue, it can increase the risk of cancer. So there seems to be a connection. We're not really sure because, for instance, for particulate matter in the air, it also increases the risk. So probably the mechanism is similar, and we think it may happen, but we don't have enough data. And since we're all exposed, it's very difficult to compare different groups of people, but probably in relationship to cancer. And the other one you wanted to know, it was the skin to cross. Oh, okay. They can cross, as I told before, what they've seen in the studies, it's very low concentrations can cross through. So like less than 1% will really get into the system, probably even less. Of course, the smaller you have, if you go into nanoplastics, probably you have a bigger internalization rate. If you go to bigger microplastics, well, even if you ingest a really big microplastics, it is unlikely that it will get through. So the lower you go, the higher the probability of internalization. But generally it's low, it's less than 1%. Of course, we're talking about being exposed to millions, billions of particles, so even 1% can mean that some are getting through, and even less will get through the circulatory system.
Okay, thank you. Final question, and then
In your dogs, several dogs, you were not able to detect microplastics. So is this a statistical issue? Is it hard to probe an animal? Or is it good news and life without microplastics is possible, and we should ask them how they did it?
This one, I have all this data from the 70s and 80s where people were using plastics. Usually they put some probes, not fluorescent probes like ours, because usually sometimes it leaches the fluorescence and we find it somewhere else and not in the microplastic, and we think there's microplastics and there's not. But for instance, radioactive probes, and I think this would be good to follow up the way it moves in the body, especially if we're trying to use more realistic microplastics with the organic matter and all those changes, the weathering, all that will change the way it behaves in the body. So I think it may be good to get some of that knowledge that we had before. Yeah.
You want to add something?
Yeah, only for to complete the information about exposure of human. Another way of exposure of microplastics and nanoplastics and airborne is the direct translocation between the olfactory cells that need a bridge between the nose and brain. So we have a direct interaction by nanoplastics and brain with no dilution or modification or dilution of contaminants that bring the nanoplastics. So for this motive we are studying the neurodegenerative effect for direct exposure to nanoplastics, environmental nanoplastics.
Right. Okay. I'd like to thank my colleagues, the presenters, and you for this excellent session. And I ask you to applaud the presenters.